so I think we'll get started. So thank you very much, everybody, for attending. So this is our 20th, which is quite the milestone. So far, we've had 62 speakers from 19 different countries, but we'd like more. So if you're interested, and you're especially in a country that we've not heard from before, please get in touch. We'd love to hear from you. So thank you to everybody that's already spoken. So on to our next lot of speakers. So today we're actually going to have two talks because somebody has unfortunately been taken ill. So we're going to have two very quick talks, 10 minutes each, and then hopefully take the discussion, or we will take the discussion into the breakout rooms afterwards. So our first speaker is Icaro, if you'd like to share your screen and kick things off. Thanks very much.
Okay, can you hear me?
Yeah.
So
I can hear, but I cannot see your screen.
Yeah.
Yeah, but what's happening with the screen? You cannot start screen share while the other participant is sharing.
Right now.
So now it's okay. Can you see my screen?
We can see that you're sharing, but it's just a dark screen.
It's black.
Can I try?
Yep.
Nice work.
That's it.
It's okay? Can you see? It's okay, my screen?
Yep, looks good.
Okay,
please
let's start. Okay, so hi everybody, my name is Icaro Caruso. I'm gonna present to you our last results about the N-terminal domain of the nucleocapsid protein from SARS-CoV-2. So the SARS-CoV-2 nucleocapsid N protein is a multifunctional RNA binding protein. This protein is composed two domains that are linked by an intrinsically disordered region, which contains the SR motif enriched in arginine and serine residues. The N-terminal domain is responsible for the interaction with the regulatory RNA sequence during the subgenome transcription, and the C-terminal domain is responsible for the protein dimerization, which is crucial for the nucleocapsid assembly. So the genome transcription of the coronavirus is a unique process because it's a discontinued process and involves the production of subgenome messenger RNAs. The regulation of the subgenome messenger RNA synthesis depends on the transcriptional regulatory sequence, the TRS, located either at the 5' end of the positive strand RNA genome, called the leader TRS, or the 5' end of each viral gene coding for the structure and accessory proteins, called the body TRS. Once the body TRS is copied, the nascent negative strand RNA is transferred to the TRSL and the transcription is terminated. The NNNTD interacts with the TRS and melts an RNA duplex formed between TRS and its complementary strands. This melting activity facilitates the template switch and plays key roles in the regulation of the discontinued transcription. So in this way, today I will present our
I think we might have lost him, unfortunately.
Something happened.
Can you hear what's happened?
Yes, we can hear you.
We can hear you.
Screen has stopped sharing.
It was my internet or it's something with the Zoom?
Probably the internet.
I'm guessing it's your internet. You want to start again? Try sharing it again.
Let's try share here again. So I stopped here, okay?
Yes, we can see that.
Okay, I will continue. So Today I will present to you our last result regarding the interaction of the SARS-CoV-2 and NNTD and NNTD plus SR with motif with the specific TRS and the non-specific DNA, as well as their activity of melting and the formation of liquid-liquid phase separation. So we start our experiment using TRS DNA as a mimic of RNA. Our first step was to measure the TRS melting activity of the NNTD and NNTD-SR using FRET experiments. We observed that the NNTD-SR has higher dsDNA melting activity than the NNTD, suggesting that the SR motif contributes to the activity. Next, we quantify the interaction of the NNTD and NNTD-SR with DNA using fluorescence experiments. First, we observe that the dissociation constant are in order of nanomolar for all interactions. And the affinity of the NNTD and NNTD-SR with the negative sense TRS, it's higher than that observed for the positive sense. We also observe that the presence of the SR motif promotes a higher affinity when compared the NNTD. In comparison to non-specific sequence, TRS presents a higher affinity, but it's not so higher that we can classify as a sequence-specific recognition. The interaction of the TRS with the NNTD and NNTD-SR presents a thermodynamic pattern, enthalpically unfavorable and entropically driven for the single strand, and the enthalpically driven and entropically unfavorable for the double strand. The same was not observed for the interaction with the non-specific sequence. And the salt and phosphate dependence of the interaction reveals us that the electrostatic interaction are important for the formation of the complex. Next, we map the residues involving the interaction with the DNAs. Our CSP profiles are quite similar to that published by Danish and collaborators using RNAs. We proceed with our analysis looking for the residues in common for the interaction of the NNTD and NNTD-SR with the DNAs. We use this representation of intersection of circles where each one contained the residues that showed CSP values higher than the average plus 1 standard deviation. From this analysis, we select residues in common for all intersections. These residues are shown in the structure, and we can see that the finger and the palm are important for the binding to the DNA. So next, we use the experimental information from the CSP titrations to build the structure models of the complex formed between the NNTD and the DNAs. In the representative structure models, the blue spheres denote the residues with significant CSP values in the primary binding site. And the cyan sphere show the remoting binding site, possibly allostatic effect. For the DSTRS and DSNS, the non-specific sequence, we observed a difference in the orientation of approximately 25 degrees, which is interesting because the TTT motif in the DSTRS is positioned closer to the beta sheet 2 formed by the beta 1 and beta 5. We also analyzed the protein DNA hydrogen bonds. We counted the hydrogen bonds with the persistent time higher than 10%. In magenta, we can see that the residues with significant count are in the same region with high CSP values. And these regions are in the N and C terminal and in the finger. some arginines and lysine residues form hydrogen bond with the phosphate groups and therefore make possibly salty breeds. The residues R92, R107, Y172, and R177 form hydrogen bonds with the DNA in most of the runs. It's worth mentioning that Dinesh and collaborators performed the mutation of the arginines R92 and R107 by glutamic acid, and they almost abolish the interaction of the NNNTD with the RNA. So next, we also analyze the energy contribution of each residues to the binding along the MD simulations. We observe that the major contribution, favor or unfavorable, comes from the positively and negatively charged residues, which are distributed in the whole protein. These results indicate that the electrostatic interaction is an important contribution to the binding, which corroborates with the fluorescence data. And interesting results happen for the NNNTD DSTRS complex, where we observed an unfavorable contribution of the TTT motif in the negative sense strand, which is close to the residues in the structural models that contributes unfavorably as well. We speculated that these results may be related to the melting activity of the NNNTD. In the end, we investigate the ability of the NNNTD and NNTD-SR to form biomolecular condensates by liquid-liquid phase separation because of their interaction with the TRS and non-specific sequence DNA. We observed LLPS only in presence of crowding agents using 10% PEG, and only for the NNNTD-SR, which indicates that the SR-rich motif is necessary for this activity. The double-strand TRS showed LLPS at protein DNA molar ratio of 2:1 and 1:1, while the single-strand TRS presented at 1:2. These results suggest that the secondary structure of the DNA can play a key role in the formation of the LLPS. It's worth mentioning that that we observed LLPS only for the double-strand non-specific DNA, and we use DAP to confirm that the base parity DSTRS was mixed together with the NNNTD-SR. And in the end, to investigate the nature of the interactions that drive the LLPS for the DSTRS, we use 10% of hexanodiol, with which promoted the decrease in the number of droplets in about 30%. And also we use 300 millimolar of NaCl that promoted the completed disassembly. So we can conclude that the NNNTD and NNNTD-SR is activity against the dsDNA. The CSP profiles for the DNA are quite similar to the RNA. The dsDNA is placed between the finger and the palm. The electrostatic interaction are important for the binding. And of course, the sequence-specific recognition for the TRS, it's not based on the affinity, I mean for the KD, but we have some indicatives of this specificity, such as the thermodynamic partner for the TRS, the different orientation for the DSTRS when compared to the DSNS, the non-specific sequence, the unfavorable contribution of the TTT motif, and we saw LLPS for all TRS and just for the non-specific duplex. So I would like to thank for all the support from the facilities and National Center of Nucleomagnetic Resonance in Rio de Janeiro, and CEMIB in the Unesp de São José do Rio Preto. And I would like to thank for all these people that work with me in this job. Okay, thank you so much for the invitation, and it's this.
Thank you very much indeed. So please save your questions for the breakout rooms, and we're going to move straight back on to our next speaker, Rob.
Hi there, everyone can hear me okay?
Yep, can hear you just fine.
Okey doke. Let me, whoops, I gotta share my screen. That's all good, and then full screen is okay?
Yep, looks great.
Okay, so good morning, everybody. My name is Rob Harkness. I'm a postdoctoral fellow in Louis Kay's lab here at the University of Toronto. So this morning, I will be speaking a little bit about my recent work on a protein known as DEGP. So what is DEGP? Well, DEGP is an oligomeric stress protective protease and chaperone in the periplasm of gram-negative bacteria. DEGP monomers, the domain architecture shown here at the top, they have 3 domains. The first one is an N-terminal serine protease domain, followed by 2 PDZ domains, PDZ1 in red and PDZ2 in green. So 3 of these associate via protease domain interaction shown here on the left to yield these trimeric building blocks as the basic functional unit of DEGP. The serine protease catalytic residues are shown here in yellow balls in the active sites, and the PDZ domains are adorning the outside of the trimer. So the very interesting thing about DEGP is that it forms a number of higher order cage structures depending on its substrate occupancy, and these are implicated in regulating its function. So in the middle here, this is the closed hexamer state where 2 trimers are stacked on top of each other. And this is mediated by 2 different sets of interactions. And so in this state, the active sites are essentially blocked from solution. This prevents substrates coming in and thought to abrogate proteolytic activity. So here, these blue interactions are between the beta sheets and loops of the protease domains, where you've got a loop from the bottom trimer coming up and plugging into the active site from another protomer in the top trimer. The other set of residue interactions here is PDZ1 interactions here, formed by these residues, and generates this hydrophobic interface. So in the presence of substrates, it was canonically thought that this structure reorganizes into these 24-mer cages shown here, which take substrates into the core of these cages. And these are actually formed by a different set of interactions formed between these PDZ1 and PDZ2 domains, where PDZ1 from one trimer is making contacts with the PDZ2 of other trimers by this set of residues shown there. So DEGP functions in the periplasm of gram-negative bacteria to maintain protein homeostasis, and it's also implicated in trafficking a number of virulence factors, and it's thought to promote bacterial virulence in this manner. So DEGP can form these cages, which can take up things like outer membrane proteins and help them get embedded in the outer membrane, as well as some of these virulence factors shown here, which come into the periplasm unfolded through the sec translocon. And so the balance between these chaperoning and proteolysis functions of DEGP is really not well understood, because you can imagine if I have an unfolded thing coming into the periplasm, why does this active protease not just chew everything up? So we were interested because of its relevance to bacterial virulence, as well as this interesting self-assembly property. We wanted to understand, well, how does DEGP do this, and how does substrate binding influence potential self-assembly in the apo state? So the 2 questions that we asked were, can DEGP adopt higher order structures in the substrate-free state? And then how do substrates influence its assembly that leads to the formation of these cages? So initially, we turned to using hydrodynamics measurements to study the DEGP self-assembly mechanism in the substrate-free state. And this is because diffusion constants are reporters of molecular size in solution, or effective molecular size in solution. So we thought that it might be a good way to examine DEGP self-assembly. we measured these things, which we call 3D DLS data sets, because we have diffusion constant on the y-axis, we have temperature on the x-axis, and then the z dimension, or the third dimension, is the total DEG-P concentration, which influences the self-assembly, which we've shown to be this range here from 11 to 200 micromolar about. So the pink is low concentration, black is high concentration. And we did this as a function of temperature because DEG-P is thought to operate as a stress protective chaperone, for example, in response to heat shock, where proteins would be misfolding. So we measured then from high to low temperature, and we see that at low concentration, DEG-P diffusion constants are consistent with something like a hexamer particle. And as you heat it, it dissociates towards diffusion constants, which are consistent with the trimer. So it seems like we have a canonical hexamer that then melts into two trimers. But the very striking thing about this data set shows that at high protomer concentration, where, for example, if DEG-P is overexpressed to perform its chaperone functions, we get deflections in these diffusion constants, which imply that it's forming very large particles, bigger than hexamers, already in the absence of substrates. And this happens right around, sort of biological temperatures where pathogens would be operative. So using a number of mutagenesis controls and some analytical ultracentrifugation experiments to sort of flesh this out a little bit more, we found that the model that best describes this behavior in the diffusion constant data set is this one here, where DEG-P actually in the apo state undergoes this complex competing pathways self-assembly mechanism, where you have path A at low temperature is the formation of this hexameric structure, and path B at high temperature is a second oligomerization pathway leading to higher order oligomers. And you can see here on this Van 't Hoff plot at low temperature on the right, the reason that path A is operative is because its association constant is tighter. As you heat, it becomes weak, and KB here is relatively temperature insensitive. So at high temperature, you get the formation of lots of big particles down the second pathway. And so this is reflected here in the fractional populations of the monomers in each type of DEG-P oligomer that we get out of fits of these kinds of models. And here, looking at low temperature and low concentration, we see mostly hexamer. And as we heat, a little bit of oligomerization, but not very much. And again, as for example, if DEG-P becomes overexpressed to high concentration, we see at low temperature, there's a bit of oligomerization, and at high temperature, a lot of oligomerization occurs, where you get a lot of these apo higher order states. And I should mention that that these studies were done in collaboration with Peter, Schuck, and Joy at the NIH, who were instrumental in elucidating this assembly mechanism. So there we go. Okay. So then, having established some of the hydrodynamic properties of this of DEG-P, we then turn to analyzing the properties of path B in more detail using solution NMR spectroscopy, just because it's very sensitive to residue-specific interactions that can inform on how DEG-P might oligomerize in this second pathway. And so we just did some very simple experiments initially using DEG-P's PDZ domains, which we suspected might be implicated in mediating this higher order assembly. So in these two plots here, we've just taken DEG-P's PDZ1 and titrated it with its PDZ2 domain, and we observed this characteristic set of chemical shift perturbations here, which map onto the residues in the higher order cage structures that are mediating these interdomain interactions. So then we went to the full-length DEG-P in the apo state and just varied its total protein concentration, and we observed a very similar set of interactions for the same residues shown here. But strikingly, when we went to analyze the fraction of bound PDZ domain interactions shown here, we find that for the individual domains, we get data sets that are very well fit by just a simple two-state binding model. But in the case of a full-length DEG-P, you can see that it's decidedly not two-state, and that there's an initial burst phase in the fraction of interactions shown here, and then it kind of tapers off here. So in order to rationalize these differences, we turn to some relatively simple models of DEG-P self-assembly here, where we've depicted trimers as these triangles, and the PDZ domain interactions can occur along the trimer edges shown with the stars. So a model in which we only have one interaction that's possible to form at each of the trimer edges can't explain these data very well because it would way undershoot the data, as well as a model in which all of the interactions are formed. For example, when DEGP forms substrate-bound cages, would way overshoot the data, being up here at a fraction bound of 1. So the models that best explain these data are this one here, for example, in red, where 2 interactions can be formed between each of the trimers, and it gives rise to this fit here. So although it doesn't do a perfect job at high concentration, it does a not bad job throughout the whole concentration range. And essentially what this means is that in the apo state, DEGP is oligomerizing into these very broadly populated ensembles where not all of the interactions are formed, and so that they're structurally distinct to these cage conformations, which are observed in the presence of substrates. So then we wanted to understand, well, how do substrates influence this oligomeric ensemble that DEGP forms in the absence of substrates? We again turned back to dynamic light scattering measurements because they're very sensitive reporters of oligomeric states in solution. We prepared DEGP ensembles, different forms of the DEGP ensemble by changing the solution ionic strength. We found that it's very sensitive to sodium chloride, and you can see here that this drives the extent of oligomerization here, as can be seen at 40 degrees. So then under each of these conditions, we just add substrates to DEGP to see, well, what kinds of cage structures do we get, and is there any effect on the any effect on the cage ensembles that we get? So with adding a little simple protein called TRF1, it's just a small folded domain, we get under all cases things that are consistent with 12-mer particles shown here on the right. And I should say that these structures were solved by Zev Ripstein, who's now professor at the University of Manitoba. And we prepared in another case, DEGP, which is bound to a substrate from E. coli. And again, under these salty conditions, we get things mostly consistent with an 18-mer structure with a little bit of this canonical 24-mer structure. So the takeaway message here is that we think that substrates are able to collapse this apo-DEGP ensemble, no matter where it lies, into cage structures which can accommodate substrates of different sizes. So we can then summarize all of these results in terms of some energy landscapes for DEGP self-assembly shown here. So to recap, we have these 2 pathways, A and B, where A is more operative at lower temperature because its energy well is deeper than the first energy well for path B. It's wider to illustrate that it absorbs more of the DEGP trimers in the form of these hexamers. But then as you raise the temperature to where DEGP might operate as a stress protective chaperone, this energy well is destabilized relative to the first energy well here. These energy wells are wider to illustrate that more trimers spill down this pathway and lead to the formation of these apo-oligomers, which are distinct from cage structures, probably having roughly two PDZ1, PDZ2 prime domain interactions shown there. And then when you add substrates to this apo ensemble, we think that this leads to the reorganization of these cages of these oligomers into cages that can accommodate a given substrate. This is reflected in this very deep energy well that's introduced on the energy landscape. It's very wide and takes up all of the DEGP trimers. So with that, I'd like to thank members of the K Lab and my collaborators who were instrumental in helping me to carry out this study, as well as the organizers for giving me the opportunity to speak here today. So thank you very much.
Great, thank you very much indeed. Okay, so let's take the discussion into breakout rooms. So you should see now